Welcome to this tutorial on blood glucose regulation, brought to you by Medical Biochemistry Power Capsule Series. This video covers the following CBME curriculum topic: discuss the metabolism and significance of blood glucose regulation in health and disease. What is blood glucose regulation? Blood glucose regulation is the process by which the body maintains blood glucose levels within a narrow, optimal range, to ensure proper physiological function and energy supply. This regulation is crucial for homeostasis, the state of steady internal conditions maintained by living organisms. This process involves a sophisticated interplay of various hormones, receptors, and enzymes that work together to either increase or decrease blood glucose levels as needed. Why is blood glucose regulation important? Glucose is a primary energy source for the body, especially for the brain, which relies almost exclusively on glucose for energy. Maintaining blood glucose within a narrow range is vital for overall metabolic balance and function. Both hyperglycemia and hypoglycemia can have severe consequences. Proper regulation helps prevent acute complications like diabetic ketoacidosis and chronic complications such as cardiovascular disease, neuropathy, retinopathy, etc. What are the key components of blood glucose regulation? Hormones. Primary hormones involved include insulin, glucagon, epinephrine, cortisol, and growth hormone. Thyroid hormones are also involved indirectly. Receptors. Specific receptors on cell surfaces, like insulin receptors and glucagon receptors, detect these hormones and initiate intracellular signaling pathways. Enzymes. Enzymatic activities, such as those of glycogen synthase, glycogen phosphorylase, and others, play critical roles in glycolysis, gluconeogenesis, glycogenesis, and other pathways. Normal blood glucose level in the fasting stage should be within 70 to 100 mg per deciliter. This level is typically measured after an overnight fast and indicates how well the body can maintain glucose levels without food intake. Normal blood glucose level in the postprandial stage should be below 140 mg per deciliter. However, it needs to be remembered that values below 70 mg per deciliter would indicate hypoglycemia, whatever be the state. Postprandial values within 90 to 140 mg per deciliter are conventionally accepted. For most normal test subjects, values are generally found to be within this range. The postprandial level is measured after eating, and reflects how well the body can manage and utilize the influx of glucose from dietary sources. The blood sample is generally withdrawn 2 hours after the test subject has taken a normal meal. An overview of the process between meals, that is, during fasting state. Blood glucose levels drop, triggering the release of glucagon from the pancreas. Glucagon stimulates the liver to break down glycogen into glucose and to produce glucose through gluconeogenesis, releasing it into the bloodstream after eating, that is, in the postprandial state. Blood glucose levels rise, stimulating the pancreas to release insulin. Insulin facilitates the uptake of glucose by tissues, particularly muscle and adipose tissue, and promotes glycogen synthesis in the liver. What are the consequences of dysregulation? What happens if the body cannot regulate blood glucose levels properly? There may occur acute complications. Diabetic ketoacidosis. It is a severe complication of hyperglycemia in type 1 diabetes, leading to acidosis and potential coma if untreated. Hypoglycemic episodes can cause immediate risks like loss of consciousness and seizures, requiring prompt glucose administration. Untreated hypoglycemia, a condition of low blood glucose, can even cause death. Chronic complications can occur if blood glucose levels are not regulated properly over extended period of time. Microvascular damage is common. Hyperglycemia damages small blood vessels, leading to retinopathy, nephropathy, and neuropathy, and many other complications. Macrovascular damage, increased risk of cardiovascular diseases, heart attack and stroke, due to chronic high blood glucose levels. Diabetic patients often suffer from some or all of the above-mentioned complications to varying degrees. depending on the extent of glycemic control. Proper glycemic control, however, prevents the development of these complications. Insulin is secreted by the beta cells of the islets of Langerhans in the pancreas. Its primary function is to lower blood glucose levels by promoting the uptake of glucose into cells, particularly in muscle and adipose tissue, as part of metabolic effects. Insulin promotes glycogenesis, that is, conversion of glucose to glycogen for storage in the liver and muscles, and lipogenesis, that is, conversion of glucose to fat in adipose tissue. It also inhibits gluconeogenesis, which is the production of glucose from non-carbohydrate sources, and glycogenolysis, which is the process of breakdown of glycogen into glucose. Overview of mechanism of action of insulin. Insulin binds to insulin receptors on cell membranes. These receptors are tyrosine kinase receptors. Binding activates the receptor's tyrosine kinase activity, leading to autophosphorylation and subsequent activation of intracellular signaling pathways, including the phosphoinositide 3 kinase, protein kinase B pathway. This signaling cascade results in a number of subsequent downstream effects. One of the key actions of insulin is to increase glucose uptake in muscle and adipose tissue by promoting the translocation of glucose transporter type 4 from intracellular vesicles to the cell membrane. Glucose transporter type 4 facilitates the entry of glucose into the cell, reducing blood glucose levels. Processes which are affected by insulin. Insulin stimulates translocation of glucose transporter type 4 to cell membrane, which increases glucose uptake. Insulin activates glycogen synthase enzyme, which increases glycogen synthesis. Insulin activates hexokinase and PFK1, which increases glycolysis. Insulin activates acetyl coenzyme carboxylase, and also fatty acid synthase. This enhances fatty acid synthesis. Insulin activates translation initiation factors and ribosomal proteins. As a result, protein synthesis increases. Insulin inhibits glycogen phosphorylase enzyme and thereby inhibits glycogenolysis. Insulin inhibits phosphoenol pyruvate carboxygenase and glucose 6-phosphatase. As a result, gluconeogenesis is inhibited. Insulin inhibits HMG coenzyme synthase in liver. This suppresses ketogenesis. Insulin promotes HMG coenzyme reductase synthesis and therefore, cholesterol synthesis is upregulated. Insulin inhibits hormone-sensitive lipase. This suppresses lipolysis in adipose tissue. Gist of the matter is that insulin increases glucose utilization, prevents formation of glucose from non-carbohydrate sources, prevents hyperglycemia, favors fat storage in adipose tissue, favors protein synthesis, and increases body weight. Glucagon is secreted by the alpha cells of the islets of Langerhans in the pancreas. Its primary function is to raise blood glucose levels by promoting the release of glucose into the bloodstream. Glucagon stimulates glycogenolysis, converting glycogen into glucose in the liver, and enhances gluconeogenesis, increasing the production of glucose from amino acids and glycerol. It also inhibits glycogen synthesis by inactivating glycogen synthase. Glucagon is the hyperglycemic hormone. It raises blood glucose level. Mechanism of action of glucagon. Glucagon exerts its effects by binding to specific glucagon receptors located primarily on liver cells. These receptors are G-protein coupled receptors, which play a crucial role in transducing the extracellular signal into intracellular actions. When glucagon binds to its receptor, it activates the associated G-protein, specifically the GS protein. The activated GS protein stimulates adenylate cyclase, an enzyme embedded in the cell membrane. Adenylate cyclase converts ATP to cyclic AMP, a second messenger. Increased levels of cyclic AMP activate protein kinase. This PK phosphorylates various enzymes involved in glucose metabolism, leading to the activation or inhibition of these enzymes. So, what are the cellular effects of glucagon? PK phosphorylates and activates glycogen phosphorylase, the enzyme responsible for breaking down glycogen into glucose 1-phosphate, which is then converted to glucose 6-phosphate. Glucose 6-phosphate is converted to glucose by glucose 6-phosphatase, and glucose is released into the bloodstream, raising blood glucose levels. Glucagon stimulates gluconeogenesis by increasing the expression and activity of key enzymes, such as phosphoenol pyruvate carboxykinase and fructose 1,6-bisphosphatase. It promotes the mobilization of substrates for gluconeogenesis, such as amino acids from muscle proteins and glycerol from adipose tissue. pKa phosphorylates and inactivates glycogen synthase, the enzyme responsible for glycogen synthesis, thus inhibiting the formation of glycogen from glucose. What are metabolic effects of glucagon? By promoting glycogenolysis, glucagon rapidly increases the amount of glucose available in the blood, providing an immediate source of energy. Through gluconeogenesis, glucagon ensures a continuous supply of glucose, especially during fasting or prolonged exercise when glycogen stores are depleted. Glucagon supports the catabolic state by mobilizing energy stores. It promotes the breakdown of glycogen and the synthesis of glucose, ensuring that the body has adequate fuel during periods of low carbohydrate intake or increased energy demand. Integration with other hormones, glucagon acts as a counter-regulatory hormone to insulin. While insulin lowers blood glucose by promoting storage and utilization, glucagon increases blood glucose by promoting release and production. Glucagon works in concert with other hormones like epinephrine and cortisol during stress to ensure that glucose is available for critical functions. Glucagon and epinephrine has synergistic effects. Both glucagon and epinephrine stimulate glycogenolysis and gluconeogenesis, but epinephrine also enhances lipolysis, providing glycerol for gluconeogenesis and fatty acids for energy. Insulin is an anabolic hormone, promoting the synthesis and storage of macromolecules such as glycogen, fats and proteins. Glycogen supports catabolic state. Epinephrine is secreted by the adrenal medulla. Its primary function is to increase blood glucose levels during stress or fight-or-flight responses. Epinephrine binds to beta-adrenergic receptors on liver, muscle and adipose tissue cells. This binding activates adenylate cyclase via GS proteins in beta receptor pathways, increasing cyclic AMP levels and activating protein kinase, which is responsible for the downstream effects. Epinephrine promotes glycogen breakdown in the liver and muscles, increasing blood glucose levels. It inhibits glycogen synthesis and stimulates gluconeogenesis in the liver. Epinephrine increases lipolysis in adipose tissue, releasing free fatty acids and glycerol into the bloodstream for energy production and gluconeogenesis. The overall impact is that epinephrine increases blood glucose and fatty acid levels, providing energy to muscles and vital organs during acute stress. Glucagon is required mainly during fasting state. Epinephrine is required during acute stress. Both ensures adequate glucose supply. Cortisol is secreted by the adrenal cortex. Cortisol is a glucocorticoid hormone and plays a key role in long-term stress response and fasting, helping to maintain energy balance and glucose availability. Please follow the flowchart depicted here. Cortisol binds to intracellular glucocorticoid receptors, forming a hormone receptor complex that translocates to the nucleus. This complex binds to glucocorticoid response elements on DNA, altering the transcription of genes involved in metabolism. What does cortisol do? Cortisol enhances gluconeogenesis by increasing the expression of enzymes like phosphoenol pyruvate carboxykinase and glucose 6-phosphatase. It promotes protein breakdown in muscle tissue, releasing amino acids for gluconeogenesis. Cortisol increases lipolysis, releasing free fatty acids and glycerol for energy production and gluconeogenesis. Cortisol inhibits glucose uptake by peripheral tissues, conserving glucose for essential organs like the brain. The overall impact is that cortisol increases blood glucose levels and ensures energy availability during prolonged stress and fasting periods, maintaining glucose supply for vital organs. Growth hormone is secreted by the anterior pituitary gland. Growth hormone regulates growth and metabolism, impacting blood glucose levels by modulating insulin activity and promoting gluconeogenesis. Growth hormone binds to growth hormone receptors, activating the JAK-STAT signaling pathway. Activation of the JAK-STAT pathway leads to transcriptional changes that support growth and metabolism. Activation of transcription of growth hormone receptor gene, insulin-like growth factor 1 gene, and suppression of cytokine signaling genes are just few examples of the effect of growth hormone in transcription regulation. This affects the synthesis of gene products, which further affects metabolic processes. Growth hormone inhibits glucose uptake in peripheral tissues, promotes lipolysis, and enhances gluconeogenesis, ensuring a supply of glucose during periods of growth and fasting. Growth hormone stimulates gluconeogenesis in the liver. It promotes lipolysis, increasing free fatty acids and glycerol in the blood. Growth hormone displays insulin antagonism and reduces the efficiency of insulin-mediated glucose uptake in tissues, increasing blood glucose levels. The overall impact of growth hormone is that it raises blood glucose levels and mobilizes energy stores, supporting growth and metabolic needs during fasting and stress. Thyroid hormones are secreted by the thyroid gland. Thyroid hormones regulate metabolism and energy expenditure, influencing glucose metabolism indirectly. Thyroid hormones bind to nuclear receptors, and then interacts with specific DNA sequences called thyroid hormone receptor elements, thereby regulating the transcription of target genes involved in metabolic processes. Transcription of many genes are regulated by thyroid hormones, including thyroglobulin gene, TPO gene, growth hormone receptor gene, etc. Thyroid hormones increase glucose utilization and uptake by cells, enhancing glycolysis and glucose oxidation. Increases expression of glucose transporter 1 and glucose transporter 4, thereby promoting glucose uptake into cells, particularly muscles and adipose tissue. They modulate hepatic gluconeogenesis and glycogenolysis, ensuring glucose availability. As part of overall metabolism, thyroid hormones increase basal metabolic rate, promoting overall energy expenditure. Therefore, thyroid hormones support increased metabolic rate and energy consumption, indirectly affecting blood glucose levels by enhancing glucose metabolism. Summary of hormonal interplay. Hormones act through synergistic and antagonistic actions. It is insulin versus the counter-regulatory hormones. Insulin works to lower blood glucose levels by promoting glucose uptake and storage, while glucagon, epinephrine, cortisol, and growth hormone act to increase blood glucose levels by promoting glucose release and production. In relation to stress response, epinephrine and cortisol are critical during acute and chronic stress, ensuring glucose availability for immediate and prolonged energy needs. Meanwhile, growth hormone and thyroid hormones play essential roles in regulating metabolism and energy balance, particularly during growth periods and metabolic adaptations, and therefore have an indirect effect on glucose regulation as well. I shall now be sharing some information related to clinical aspects of blood glucose regulation. Dysregulation is mainly seen in diabetes mellitus. Type 1 diabetes mellitus. Type 1 diabetes mellitus is an autoimmune disorder where the body's immune system attacks and destroys the insulin-producing beta cells in the pancreas. This destruction leads to an absolute deficiency of insulin. With little to no insulin production, glucose cannot enter cells effectively, leading to elevated blood glucose levels, a condition known as hyperglycemia. The lack of insulin also fails to inhibit gluconeogenesis and glycogenolysis in the liver, further raising blood glucose levels. Unchecked lipolysis results in the overproduction of ketone bodies, leading to diabetic ketoacidosis, a potentially life-threatening condition. Type 2 diabetes mellitus is primarily associated with insulin resistance and a progressive decline in beta cell function. Risk factors include genetics, obesity, sedentary lifestyle, and poor diet. In insulin resistance, target cells such as muscle, liver, and adipose tissue become less responsive to insulin, requiring higher levels of insulin to achieve the same effect on glucose uptake. So compensatory hyperinsulinemia occurs. The pancreas initially compensates by producing more insulin, leading to hyperinsulinemia. Over time, beta cells become dysfunctional and fail to produce adequate insulin, resulting in chronic hypoglycemia. Type 2 diabetes mellitus often coexists with other metabolic abnormalities such as hypertension, dyslipidemia, and central obesity, collectively known as metabolic syndrome. The diagnostic criteria for hypoglycemia conditions vary slightly between the World Health Organization and the American Diabetes Association, but they generally involve measuring blood glucose levels. Here are the key points related to World Health Organization criteria. It is diabetes mellitus when fasting plasma glucose is greater than or equal to 126 mg per deciliter, or when the 2-hour plasma glucose during an oral glucose tolerance test is greater than or equal to 200 mg per deciliter, or random plasma glucose, in the presence of classic symptoms of hyperglycemia, is greater than or equal to 200 mg per deciliter. It is impaired fasting glycemia, when fasting plasma glucose is between 110 and 125 mg per deciliter, and when the 2-hour plasma glucose during an oral glucose tolerance test is less than 140 mg per deciliter. In impaired glucose tolerance, fasting plasma glucose is less than 126 mg per deciliter. The 2-hour plasma glucose during an oral glucose tolerance test is between 140 to 199 mg per deciliter. Here are the key points related to American Diabetes Association criteria. It is diabetes mellitus when fasting plasma glucose is greater than or equal to 126 mg per deciliter, or when the 2-hour plasma glucose during an oral glucose tolerance test is greater than or equal to 200 mg per deciliter, or when the glycosylated hemoglobin is more than or equal to 6.5%, or random plasma glucose in the presence of classic symptoms of hyperglycemia is greater than or equal to 200 mg per deciliter. In this criteria, both impaired fasting glycemia and impaired glucose tolerance have been clubbed together under a single category, which is prediabetes. It is prediabetes when fasting plasma glucose is between 100 and 125 mg per deciliter, or when the 2-hour plasma glucose during an oral glucose tolerance test is between 140 to 199 mg per deciliter, or when the glycosylated hemoglobin is between 5.7 to 6.4%. Clinical presentation. Symptoms of diabetes include polyuria, which refers to frequent urination; polydipsia, that is, increased thirst; polyphagia or increased hunger; weight loss, fatigue, blurred vision and slow-healing wounds. Diabetic ketoacidosis presents with nausea, vomiting, abdominal pain, and altered mental status. Complications, such as cardiovascular disease, neuropathy, nephropathy, and retinopathy, may also develop in both types. What about hypoglycemia? It is also a clinical disorder related to abnormal blood glucose regulation. Hypoglycemia is the opposite condition. Hypoglycemia occurs when blood glucose levels fall below normal ranges. Causes include excessive insulin administration, inadequate food intake, excessive physical activity, alcohol consumption, and certain medications. Impaired glucose production is another cause. Insufficient glucagon and epinephrine response can fail to trigger glycogenolysis and gluconeogenesis. Symptoms include tremors, sweating, palpitations, anxiety, confusion, dizziness, and, in severe cases, seizures, loss of consciousness, and death. Prompt recognition and treatment are essential to prevent serious outcomes in the short term. Hypoglycemia is considered more dangerous than hyperglycemia. A blood glucose level below 54 mg per deciliter is considered to be very dangerous. Diabetes mellitus presents a multifaceted health challenge, encompassing a broad and elaborate field of study. The introductory remarks provided herein merely scratch the surface of diabetes mellitus as a topic. For a deeper exploration, a dedicated and comprehensive tutorial on diabetes is accessible in this Medical Biochemistry Power Capsule series, where you can learn about the multifactorial nature of this disease, including genetic predispositions, environmental influences, lifestyle factors and other aspects.
Thanks for watching this video. Our next tutorial in the Medical Biochemistry Power Capsule series is titled Diabetes mellitus. The tutorials presented in the MBPC series are not intended to be a substitute for your textbooks. However, we feel that these tutorials can prove to be very valuable as complementary educational aids and for purpose of rapid revision prior to exams. If you have enjoyed this video, don't forget to hit the like button, subscribe for more exciting content, and share with your friends. And do keep watching our channel to remain ahead in the game.